All right, well, thanks everybody for coming. My name is Jessica Cox, and this is my colleague, Corey Harper. We'll be presenting together. We're from Elsevier. I'm not sure how many people of you are familiar with the company, but it's traditionally known as a scientific publishing house, and we've now recently transitioned to more of a data analytics company. And we work in the labs division, so we do a lot of research. So today we're going to present to you some of the things that we work on and we find interesting at the company, and some of the tools that we actually use to do it. So I'll first ask, how many people here have written a paper before? Okay, so enough of you. You know what the process is like. So I'll try to put yourself in the shoes of a scientist now, somebody who's collecting a lot of data, writing up this manuscript, submitting it, and then now you want some feedback. Was your work any good? Are people interested in it? So this can come in the form of getting emails, could be going to conferences and talking to your colleagues and seeing what they thought about it. But really, what most people are waiting for is how many times it's cited. This seems to be the gold standard in a lot of fields. How many times are people citing your work? But we're not really sold that that's the best measure of how people value your work. So citation networks, I'm not sure how many people here are familiar, but they're a pretty old problem within the fields of scientometrics. And this is just one example of a way that you could dice up visualizing a field and how people are citing one another. So this is from an article back in 2010 that essentially is just looking at how many times scientists are citing one another within the field of stem cell research. So that big orange circle is out of Dr. Yamanaka's group, who won the Nobel Prize in 2006 for discovering a new way of inducing cells to become stem cells. So you can kind of get a visualization of these different groups that have evolved, who's citing who, and it wouldn't be surprising if you found out that they're all working on different areas of stem cell research. But again, if you just count all the cites, is that really telling you everything? If everybody cites you and says, this sucked, this was bad, that's good to know. You want to know what people are saying. So this is kind of what we find to be a very interesting NLP problem, and what we're going to be talking about today. So just to show one example, that big orange circle, their main paper out of there, these are two examples of how it was cited. One time it showed up in the materials and methods section, so it's being referenced specifically as an important method. And even if you were to see the title, I know it's kind of hard to see, but it is a methods paper. And this other paper just referenced it as background information, just general information about the field. But if you were to look, there's very different language used when you're referring to some of these things. So this is the problem that we want to explore today. So today we're going to kind of hit this sweet spot in between an analytical talk and a technical talk, talking about some of the tools that we use at Elsevier, specifically Pyspark and Databricks, to do NLP of millions and millions of journal articles to understand how they're being cited and talked about.
Thanks, Jess. So Wim actually went through a bunch of the stuff we were going to talk about here about Spark, sort of foundational Spark principles, so we can save a little bit of time on that. But we don't use Spark streaming much at Elsevier, but we're really invested in the Spark architecture itself. We have huge amounts of data that we're constantly trying to process and analyze and make sense out of. This comes in the form primarily of XML. We're a scientific publisher. We have a database of something like 15 to 20 million scientific articles that we've published over the years, and so that's the input that we're typically working with here. Now, we're going to introduce a tool called Databricks, which we use really heavily, and what Databricks is it's kind of a Spark management platform, and I'll show you what that means a little bit later, but it gives you the ability to control your clusters through a web graphical user interface and do your work in a notebooking environment there. And the great thing about this notebooking environment is that it feels a lot like working in Jupyter. It's based on Zeppelin, which comes with Spark out of the box, but because Databricks is in a cloud environment, you can share these notebooks back and forth, and you can see each other's cells and the execution of them and the results, and you can even, if you've permissioned it to allow it, you can even run each other's code at the same time in real time. So it's great for collaboration. The other cool thing about it is it offers a free community edition that gives you a single cluster or a single node cluster of 6 GB for free that you can play around with and work with. And we're going to give you some notebooks and some data that you can play with there. I can skip over this because you saw it in the last talk. I'll talk really briefly because I know some people came in fresh, but The idea of Spark is that it's a driver executor environment built on MapReduce, where the driver loads up and distributes work out to executors, and then those executors load up data and perform computations and send it back to the driver to compile and make sense out of. And what's really compelling about this is that it's a delayed execution environment. So you have transformations on the one hand and actions on the other hand. Transformations are changes to the data, additions to columns in a table, computations that happen on it, but they don't actually compute until it gets to an action, something that you're doing with that data, either saving it to a disk or displaying it. And what's great about that is it allows you to queue up a whole bunch of transformations, and it optimizes the execution plan for that set. And so you can build these Spark pipelines that allow you to do that pretty effectively. So in our case, because we're talking about these citation networks, we've built up a data set that we're going to share with you. And I'm not going to get into the code for the left side of this, in part because it's all in Scala and this is a Python conference. But what we've done here is we've taken as seeds about 175 Nobel Prize winning papers going back to 2005. And we've collected every paper that cites them that Elsevier publishes. Then we filter that to about 250 papers that are in a data set that we call CC BY, which is Creative Commons attribution license, which means anybody can download that data and do anything they want with it for free, so long as they sort of link back to us. So we give you that data set, and we're gonna take all of the citations in the papers that cite these 175 papers, whether or not that citation is citing the paper or not. And we're going to filter those by a all science journal code system that we have at Elsevier that categorizes the journals that these papers are published in. So we're limiting it to the biomedical domain. And the end result of this is 30,000 sentences that we're going to be able to give you as a takeaway in a CSV file to work with if you want to sort of play around and learn more about this architecture. A quick note about the features that are in that data set. The IDs of the articles, that all science journal classification code, the age of the citation, so the difference between the Nobel Prize paper being cited and the paper citing it, the section of the article that it came from, so whether it's in the review section or a review article or the method section or the introduction, the article type, what it is it a conference paper, is it a journal article, and then the text of the citation, and that includes the sentence that the citation is in, the sentence before it, and the sentence after it. So with that, I'll sort of jump over into that Databricks environment and show you how that works. So real quick, just sort of intro to what I mean when I say it's a Spark management environment. It gives you a GUI to be able to spin up Spark clusters. You name it, you can pick the version of Spark that you want to work with there and create it. I'm not going to do that right now because it takes a little while for it to spin up, but I've already got one running here for this context, right? Another thing that you can do is you can then load libraries to this cluster. So you create a library and you can load up Java jars or Python from PyPy or uploading an egg, and it will attach it to the cluster so you can import it. So things like NLTK or SpaCy that you might want to work with, you can add here. Finally, you can load up data. So you can read in, you can read in data files from your system, from your hard drive, and just upload them, and it will put them on that Spark cluster so you can work with them. You can also mount S3 buckets and do various other things. Again, not going to do that right now because it takes some time, but we've kind of pre-done all of that, and I'm going to bump the font up a little bit so we can walk through this and show you what that actually looks like and what you can do with it. So you load up a data frame. So the last speaker talked about RDDs, which is sort of the base level API that Spark gives you for working with these resilient distributed data sets. On top of that, there's a data frames API that is a lot like what you'd be familiar with if you've used Pandas data frames or R data frames. And so that's what we're loading up and working with here. We're going to load this CSV into that data frame, and you can start just looking at it, doing some base level analysis. So you display it and you see indeed those same features that we were talking about, right? The ASJC codes, which are maybe more than one per article. The ID, the sentence that the citation is in, the previous and next sentence, et cetera. You load up a bunch of the default miscellaneous Spark libraries that you might want to be working with. And then you can say, count the number of rows in that data frame. About 30,000. You can count the number of distinct values in a particular column, so 250, right? And those correspond to the numbers I was giving you before. 30,000 sentences, 251 papers. So we're going to do a little base level analysis on this. We can take, we're going to get rid of null values, and the reason we have to do this is because not every sentence has a previous or following sentence, because we're limiting those to sentences within the same paragraph. So if the citation is in the first sentence of a paragraph, we have a null value for previous. And some of the analytics in NLTK will blow up if you deal with that. So we're just going to fill in those nulls with blanks. And we're going to concatenate a full text column. And each time we make one of these changes, you just add a column to that data frame. It's a transformation. It's queuing up a concatenation of those 3 sentences and adding a new column to that data frame. But it's not actually acting on that until you do something with it. Like this, like displaying it. That action would affect the thing actually happening. And you see that full text column, and you see that we've got those 3 sentences together, complete with the citation itself, which is a little bit noisy, a lot of punctuation, other noisy things. So we can start using sort of basic cleanup pipelining. Create a stripped, well, we had some URLs in some of these papers that were making our tokenization a little squirrely, so we used a regex to strip out URLs. We used a regex to strip out the citation values themselves, the authors and years, which aren't providing a lot of information in this context. We're going to lowercase our data. We're going to strip the punctuation. We're going to make 2 and 3 space separators into a single space. And you've got this nice cleaned up raw text column. And we can start digging into our features here. But in order to do that, we're going to want to select the features that are relevant to us, right? So we're going to just pull that subset of features, including the cleaned up ones, that we included in the feature list that we were talking about before. And to remind you of what those are, there they are. Now, this ASJC code system is hierarchical. So there's a default hierarchy to the way these numbers work. So we want to take the first ASJC code for these articles and then bump it up to its top level place in the hierarchy. And what we want are ones that fall into these 5 categories, because these are the biomedical and life sciences articles that we want to look at as a subset of this Nobel Prize corpus. So we can, again, easily do that by first splitting on an internal delimiter in that column and grabbing the first ASJC code associated with the paper, and then just taking the first 2 characters of it and mapping it back to those categories and doing a filter on it. And we're going to add those to our data so that we have that data in here to look at. I think with that, I'm going to hand it back to Jessica for a little bit.
Sure, so now we can actually start visualizing some of these features. So one of the nice things about Databricks is it allows you to do really quick visualizations. So this is actually a table, and very quickly and easily you're able to push it over to a histogram. It's not perfect. You can see that not all the indices match. You're not having everything marked. There are different things that it's not as flexible as something like matplotlib, but it's also easy to import, and you can also make your own. But if you're doing some quick exploratory analyses, we've been very happy with using this just to get a sense of what we're looking at. So you can see that the age of the citations is pretty right skewed, which is what we would expect. And if we're looking now at the location of a citation within a paper, we also see that that tends to be skewed. So we only looked at section title for the article types. Anything outside of article types will probably have very detailed section headers that are very pertinent to whatever the review is on. So if you mouse over this, you can actually see the introduction had the second most amount of sentences within it behind reviews. We made them their own category, and then again, looking at other miscellaneous sections and so on. So we see some skew again by some of these features. I'm not gonna go through every single one. We also see with ASJC codes, mostly with the higher level ASJC codes. So just as we were expecting, the majority of these sentences are falling within this one category that really captures a lot of the molecular and cellular biology work that's going on. I won't go through every single one of these. A lot of these are collapsed, so again, if you're interested in playing with this notebook, it will be available. Feel free to expand everything and go through, and you can get an idea of what all of these things look like. So now we can actually start doing some of the NLP And we used NLTK for a lot of this to dig into it. And we're really just going to look at some basic things, n-grams, bigrams, trigrams. We're going to do it by feature type to see if there's anything that's really sticking out where we can get a sense of how people are talking about this entire corpus of sentences and what seems to be any of the patterns. So we just start by importing our NLTK library, followed by some more specific libraries. So here we're importing stop words as well as bigrams and trigrams. What's really nice is here we can just add to our stop words list, specifically some domain-specific terms such as et and all. They're not really going to help us, and they show up a lot for anything that may have slipped through. If you were to do more domain-specific chemistry, whatever it is, this could be easily tailored. So we're going to look first just looking at these sections. So this is just making a list. This command is just making a list of all the distinct section headers that we see within the data frame. And this cell is actually just a for loop. And it kind of wrapped around on itself. But it's essentially just a for loop where it's iterating now through that entire data frame and looking against that list. And what it's going to do is just print the section title as well as how many sentences were within it, and then apply the frequency distribution function within NLTK to return the top 20 most common words for all of them. So this is what the output for this would look like. We see materials and methods. There's lots here. I'm not going to go through the whole output because it is pretty long, but we pulled some things out. We saw that within materials and methods, words like described and previously and used came out. These are things that we would expect. So people are referring to previous protocols. In discussion and results, we see references to other studies, may, shown studies. In introduction, we see more of these higher level concepts, more background information. Again, things that we all expected to see. If you were to look at that output, a lot of the things that come through are random letters or numbers. So this is one of the fun parts of working with biomedical literature. These are all kind of tokens of gene names. There are probably different ways that we could approach it for, but for something like this, we wanted to do a quick analysis and it was fine. We actually saw a lot of gene names come out. Then we can reuse that code to actually look at bigrams. So again, instead of now applying just the, we're applying the bigram function as well as the frequency distribution and returning the top 20. So again, we're going to look by section. And it's a little bit more of the same, but now we're seeing an extension of that, previously described or kindly provided. So people are referencing other materials they may have gotten. Discussion and results, we see specific references to one study, a mutagenesis study. And again, introduction is kind of more of the same. So we did this for trigrams. I won't go through the whole thing, but again, it was more of the same. So this was really encouraging. We're seeing that there are specific patterns in the way that people are talking about the papers that they are referencing it, just what we hypothesized. And Corey has some visualizations of that.
So one of the things that we do a lot in our workflows at Elsevier, we'll have a pretty large-scale batch work that we're doing with like millions and millions of articles and tens of millions of sentences, and we'll want to pull out subsets of that and kick them out to other systems to do further analysis. That's a little bit harder to do within the Spark environment, right? So like visualizations, we might want to load things up in Jupyter or another tool. We do this sometimes with loading things into Gephi to do network visualizations of these citation graphs. But here I've just got a real basic example where I'm using the same code Jessica just showed you, but instead of displaying the bigrams for those sections, I'm going to collect them into a JSON structure that kind of makes sense for working with one of the templates that Mike Bostock published for doing D3 visualizations in blocks.org. Right, so we collect that up and we do the same thing, but we build this JSON file, and then we can dump that JSON file to the file system. And because we're putting it in this file store on Databricks, we can actually download it and load it up into blocks through a gist. And all this is it's a zoomable bubble chart of these bigram frequencies. And it's the exact same stuff that Jessica was just showing you, that in the materials and methods section, the bigrams that typically show up, and you can't really read that there on the resolution on this screen, and I'm sorry, but I'll tell you that the big ones are previously described and described previously. Whereas in the introduction, stem cells and transcription factors are the most common things that get talked about. Right, so it's really easy to be able to take these subsets of data and kick them out places. The other thing that I wanted to show you quickly is in addition to the kind of inline display stuff that Jessica was showing you before, it's really easy to use like Matplotlib, or if you're a R person, ggplot, right within this Databricks environment. So I've loaded up, Andreas Muller, one of the maintainers of scikit-learn, has a GitHub for a library for word clouds. And I loaded that up, and I just used one of his default examples, but instead of loading files from the system, I load up the whole text list of all the words in our entire corpus of text and spin up a word cloud of it. And you see stem cells and grid cells and place cells. And one thing that's important to point out here is that you can't just say plot.show the way you would in Matplotlib in Jupyter. You have to wrap it in this display function to get that plot to display in Databricks.
All right, so we did this for all the other features. Spoiler alert, nothing else was as exciting or interesting as section title, which was not really that surprising, right? We would know that people, again, are referencing things very differently they would in a materials and methods section versus a discussion. So now we're going to move into some LDA just to wrap up. So this is where we use the MLlib pipeline. And I really enjoy using this. It's very easy to chain things together. And as Corey said, all you're doing is chaining together a bunch of transformations before you're actually executing anything. So here, just importing some of these basic libraries, we tokenize all the text and remove all these stop words. And again, just to point out, it's pretty simple code. You're inputting a column and setting the output column and chaining everything together. We did TF-IDF on these. We use the count vectorizer. That kind of acts as the term frequency builder. And then we use IDF, inverse document frequency, on top of that. And we come up with this variable rescaled data. And that's where we perform LDA, or latent Dirichlet allocation, so just a probabilistic model for topic generation. We chose 10 topics, thinking, we might get something interesting out of that. And again, you can see it's relatively easy to generate. And what this code is going to do is generate a data frame down here where we have the term indices, the term weights, and let me see if I can scroll over. This is what happens when you don't use your own computer. But you can see, you can see some of these topics that come out. So we saw things relevant to mouse studies. We saw things that were more specific to particular gene and protein names. So we did see some of these things that began to come out. If we were really serious about pursuing this is where we could go back up and play with some of these parameters, because this only took about a minute to run. Which was really nice, or not even, 22 seconds, which was really nice. We compared this to just using GenSim and NLTK and doing the same exact thing, running LDA on 10 topics, and it took almost 15 minutes. Let me see, down here. So we're not going to get into the comparisons of what the topics are. It's not important, but this is just something to consider. If you want to play with your favorite NLTK and GenSim libraries, go ahead, but I personally came away from this much more preferring the MLlib stuff. So let me go back to the slides quickly just to wrap up. So we wanted to see that original paper. Was anything really jumping out at us? And so what we did was we boosted our data. Sorry, we can't share this all with you because they weren't all from CC BY papers. But we got about 2,400 sentences that we're citing. Again, I apologize for the poor resolution, but this is just to show the distribution of where it's being cited, mostly within the introduction and the results and discussion. And just looking at some of the sections and the n-grams we saw previously described, again, it keeps coming up, but this is good. So these are things that we can now pursue if we were going to start building a citation network and understanding how this has kind of shaped the field and what people are really thinking about it. We see reference to a specific treatment too in a trigram. So a lot more of the same. So just to quickly wrap up, we make the argument that there's way more to consider than just the amount of times your paper is being cited. As I said, if everybody's disagreeing with what you did, that's important to know. Or if people are holding it up as some groundbreaking discovery, you need to know the language that's surrounding that. But you also can't be expected to read the 500 papers that cited your own, if you're lucky enough to get cited that many times. And NLP is what we're using here to actually allow us to understand how and why your paper is being discussed. And Databricks and Pyspark for us is a really nice platform for us to kind of go through this. And as I mentioned, we're looking at millions and millions of papers at a time on a scale much larger than this, and it allows us to really go through all of that. There's lots more things that you could do. We wanted to do some k-means clustering. We wanted to, you could go into Word2Vec and Sense2Vec to get a better sense of some of the things that are coming out, vectorizing these terms. Parts of speech parsing. This is something we're starting to look at and understanding if there's more patterns there that we're missing, more general patterns we can pull out. And so for those of you who aren't stem cell researchers, you can think about this in your own lives. How are people, talking about the links that they post on Facebook? Again, how are they referencing some of these things? You could do sentiment analysis. We know for us, scientific papers, sentiment analysis is not very helpful. So it's not something that we performed, but again, it's something you might consider doing. And for just a more general application, looking at categorizing some of these papers or categorizing, say, newspaper articles or things like that. So for you guys, we have a few take-homes. That notebook that we showed will be available to run, and you can run it on Databricks Community Edition, as well as the data set, which will be in a CSV format. So have fun, 30,000 sentences if you like NLP. And we also have a link to the archive version of the notebook. And everything is up on our Mendeley data set. You'll see everything there. Go ahead, take a picture, and have fun playing with it. And with that, I guess we'll just take any questions if there's anything. Thanks for listening. Sure.
Two questions.
Sure.
Have you considered citation suggestion? So you read through a paper that's going to be edited and say, well, our algorithm said, there are some other papers that have similar wording.
Yeah, that's something we're very interested in.
Also, have you thought about reverse engineering papers where actually you say, the opposite of that? Okay, I know I read these papers and I have this data. Do you generate a first draft of the paper for me?
Do you want to come work for Elsevier? Yes. These are all things that we've thought about, and really just building tools to make writing a paper easier for scientists. And whoever's writing the paper, but those are great ideas.
We're also thinking about things like hypothesis generation, right? Like looking for gaps in the literature where you have a citation that accentuates that, this is an open question. Maybe you can suggest authors that this is areas that they might want to do research in.
Yep.
Have you done any scoring of positivity or negativity of the words around the citation and then try to take the temperature of the...
Yeah, we have in the past. There's some work done on that, and nothing really interesting comes out. I would say most scientists tend to speak pretty neutrally, with the exception of saying they support or don't support a piece of work, but it's very small in the scheme of all of the sentences that are being presented. But okay, well, thank you so much.
And we'll be around today and tomorrow if you want to come chat with us at a break at some point.